assignments. We zitten al live op YouTube, dus dat je het weet.
Oh, okay.
2 people watching. Oké. Kan je best nog even op mute zetten als je dat wil.
Ja.
Ready? Je kan me goed horen?
Ja?
Mooi. Okay. There we go. Okay, hello everyone. Victor and I are hosting a couple of Q&A sessions. We're in this first hour until 2, Victor is going to be talking about the questions that you posted online in the discussion board. And maybe afterwards there is still some time left to be talking about our questions that you might have. So Victor, I'm basically admitting everyone and making some textual notes. So I'm hosting. The floor is yours.
Okay, thank you, Ilya. Okay, so the first question was by Manala Layer, not exactly. What are the 3 most important features to distinguish apperceptive, integrative, and associative agnosia? Well, I don't know whether there are 3 most important features to distinguish, but the most important features of each of these 3 conditions are summarized for each condition in slides 21, 26, and 31 of the lecture on agnosias. And of course, after those slides, this list of features is further explained in the slides following. So, for example, if we take let me see, can I share my screen? How does that work? Yeah, share screen, that's this. This one. Can you see this, Ilya, the screen?
Yeah, I can see it.
Okay, so for example, here on slide 21, this very simply lists the features of apperceptive agnosia. So it's mostly right hemisphere. It's mainly a problem of perceptual organization, and then it also talks about these different tasks at which these people are typically bad. So the calling picture tasks, they're bad at copying, they're bad at these unusual view tasks, which are of course explained in the following slides. So they are left on the left, you see the calling picture tasks in which they are worse than normal people are. You see here some deficits in copying. This is the unusual view tasks where you know, people may still recognize that this is a stapler, but in this unusual view, you know, it's very rare that, of course, you look at a stapler only from this angle. Then, of course, it's also for us more difficult to recognize, but for these people, it's then even worse. Yeah, so that's the idea of this. And same with a task like this. These kinds of patients can or cannot, you know, of course, with patients, you always have all sorts of differences between patients, but in this shadow test task, you'll see that they may still be able to recognize these heads if they are in a normal lighting, but if they are in this extreme shading type of lighting, of course, we too would have difficulties recognizing it, but they have even more difficulties. So anyway, which makes the objects more difficult to recognize, then you will see that these patients really fall apart. Okay, so of course I'm not going to go over all the other different agnosias, but that's more or less the idea. So you have to know the features of the condition, like here in apperceptive agnosia, or like here in integrative agnosia. And you know, this is just a list of the features, but the features themselves are explained in the other slides. Same here for associative agnosia. So I hope, Mala, that this answers your question. If not, maybe you can signify us in some way. I don't know how that works, but is Mala there? Okay, well, if not, then I think I should go on to the.
I'm just going to unmute everybody for just 1 second, and please don't start talking all at once. But I just want to know where everything is clear. So if you have a question, please say. Any questions?
That works, but
I don't hear anything, so I'm going to mute you again. Okay, then we're going to go to the next sorry, 1 second. Then we're going to go to the next question. So there's different features that are important distinguishers between different forms of agnosia, and you need to know which features.
yeah, right. So then the next question is, in the recorded lectures from last year, Ilya and Victor said that Victor's topics, there was no need to read the book. Does this still apply for this year's exam? Short answer is yes, that still applies. So that's if you know everything that's in the lecture slides and understand that, you will be perfectly fine. Simple, I hope. Okay, next question. Noah Klassen, how does the saltatory conduction work? All right. Now I understand that this is indeed. Let me go to the slides. Well, this is a PowerPoint version next to the other one. Is this shared again? Wait a second, I'll share it again.
Now I see you.
This one. All right. Saltatory conduction. Well, in order to understand saltatory conduction, you first, of course, have to understand the normal way in which the action potential proceeds over a neuron, which is explained. Let me see, what is that? Which is explained here. So, of course, at some point you get this depolarization of the membrane potential, which is caused by the influx of sodium. And then because of these sodium voltage-gated sodium channels, you get even, because of the depolarization, these open, and then you get even more sodium flowing in, and then, you know, this is this Hodgkin-Hochuli cycle. You get a very rapid depolarization of the membrane potential, and this is what we call the action potential. Now, of course, if that happens at a particular region, say over here, you get depolarization there, and then, of course, these depolarization spreads over the remainder of the axon, so that now at the neighboring region, again, you get the opening of these voltage-gated sodium channels, and you get more depolarization, etc., etc. So that from there on, this action potential will spread like this over the axon. Now, of course, this happens at a particular rate. The rate depending on how fast these voltage-gated sodium channels open. How fast these sodium channels open at each successive site, and that takes a particular amount of time. Depends also on all sorts of variables, like the amount of voltage-gated sodium channels, the thickness of the axon, and all sorts of other things. But this gives you typically speeds like a meter per second or something like that. So it's fast, but not really all that fast. Now, in saltatory conduction, which is explained here, what you have is, because of these insulators, which are these Schwann cells, they provide an insulating region around the axon. The depolarization that you have here can now spread at once from this region to this region, and so it goes from here, and then there's electrical conduction from here to here, so that now you have a depolarization, opening of voltage-gated sodium channels at this location, and then you get depolarization then, and then this potential difference then spreads electrically again to this region, etc., etc. So you get these, that's why it's called saltatory conduction. You get these jumps, and so you get this opening of voltage-gated sodium channels here, and then almost instantaneously you get it here, because electrical conduction, of course, is instantaneous. So if there is a potential difference here through the extracellular fluid, which is a conductor, you'll get almost instantaneous also that this electrical field is also noticeable, or is also, it can also be depolarized the membrane here, and then you get opening of voltage-gated sodium channels, and then you get the whole process again. So what happens here is that you can skip all the voltage-gated sodium channels which are beneath this insulator, and that's why you get much faster conduction with this saltatory conduction than you would get with a situation where the opening of voltage-gated sodium channels each time has to wait, for the next one to open. And so that's why saltatory conduction is generally much faster than normal progression of the action potential over the action. And now you can reach speeds, well, up to 150 meters per second, which is of course very important. For example, if you want to send a signal to your feet, so if that would take a meter per second, you can only react in 1.5 seconds, say, to kick a ball or something. So that's why this is particularly important, this saltatory conduction, in peripheral nerves, and nerves going from your brain through your spinal cord to your muscles. That's where you will typically find these Schwann cells, providing this myelin sheet surrounding the axon. Something that's often misunderstood is that electrical conduction, because that's very important to understand it, is that electrical conduction, so say if you have a potential difference over here, this is noticed almost immediately, because electrical conduction is of course at the speed of light, is noticed almost immediately also over here. And so that's why you can go very fast from here to there. Maybe that's a note that is something that should be noted.
I think everything is clear. Any other questions? You can post that in the chat, and I'm posting some answers on the discussion board as well, as Victor is talking right now and explaining. I give you a couple of seconds to say where everything is clear or not, else we're going to continue to the next question. And I think that you briefly explained the membrane can be seen as an electrical circuit with resistors and capacitors. Is this necessary to know for the exam? And if so, could you explain this one more time? This is something that is not in the exam, right, Victor?
Yeah, I already answered this on the Canvas site. Yeah, you don't need to know that. It's just if you are I don't know, if you're into these kinds of things, maybe of interest. But it's and a detailed explanation is given on the link below slide 25. And so on slide 25 there is a link which explains this electrical circuit analog. But indeed, you don't have to know this. This is just if you like that.
One other question related to the previous question that I got through the chat. What about people with MS? Is their myelin sheath damaged? What happens?
Yeah, this is exactly one of the problems of multiple sclerosis, which is indeed on slide 37. It's an autoimmune disease where the immune system destroys this myelin sheath of neurons. And as you can understand, if the myelin sheath is destroyed, the saltatory conduction is no longer working properly. So the conduction speeds will be much slower. And of course, that gets you into all sorts of trouble properly controlling your muscles. And that's what's also expressed then in the symptoms that you see. So people are typically have muscle weakness. They have all sorts of problems with coordinating their muscle movements, which is, for example, expressed in what's called dysarthria, so that people have very difficult to understand speech. Dysphagia, so that they cannot swallow anymore, because swallowing, of course, is a very coordinated muscle action where all the muscles have to do the right thing at the right moment, because otherwise, it goes all wrong. So those are typically things that you will see, but you also get visual problems and all sorts of also more say central problems, as they're called. So things which are going wrong, because of course also in the brain itself there is a role for these myelin sheaths. But it's mainly initially what you will typically mainly find are these problems with all sorts of things which have to do with coordinating your muscle movements, and not only your skeletal muscles, but for example also the muscles of your urinary tract or your bowel and things like that.
Okay, great, thanks. So the next question is, what are the ocular dominance columns and the related information about orthogonal organizational components of V1 plus the hypercolumn?
Yeah, okay. Do you now see this other? I don't think so. Okay, so I'll share that. Yeah. So this is the ocular dominance column is coming from the fact. Where do we see that? I guess it's best shown here. It's coming from the fact that each of the two eyes project to different parts of primary visual cortex. And so this is where the right eye projects to primary visual cortex. This is where the left eye projects to primary visual cortex. If you go back a little bit here, you may remember that each of the two eyes, so for example, this part of this eye projects to this part of the cortex, but the same left side of the eye of the other eye also projects to this part of the cortex. So each hemisphere, each part of the visual cortex has input from two different eyes, left and right eye. But these two different inputs remain segregated at first, particularly so because they project to different parts of layer 4. And then, of course, this layer 4 projects to layers 2, 3, and to layers 5, 6, which are here below. So also these layers then, of course, primarily receive input from this one eye. Same here for the other eye. Now you can visualize these ocular dominance columns because, they're called columns because they are a column through the visual cortex. So what you see here is a sheet of visual cortex, and then you have these columns. You can visualize these with a technique which we call desoxyglucose labeling. Basically what you do there is you inject a tracer in one eye, and then this tracer will be taken up by the neurons and end up in this case, where these nerves project to. So in this case, the one eye that's been injected to, and then you can visualize this with an immunostaining. Those are all details you don't need to know. But anyhow, you can visualize the input from the one eye, which is shown here in black. And as you can see, they have these. Yeah, should we call them columns or what are they? But they are columns in the sense that they run through the cortex in a vertical fashion. So this is where the one eye projects to, black is where the one eye projects to, white is where the other eye projects to. In this image, you're looking on top of the visual cortex. So that's the ocular dominance column. Now, another anatomical functional parcellation in the visual cortex is what we call these blobs. So you have, this is a peculiar feature of visual cortex. If you stain for this enzyme, cytochrome oxidase, it's just an enzyme that's used for metabolic activity. But somehow in primary visual cortex, you get much more staining, much more. The enzyme is more abundantly present in these small regions, which we call these blobs. In primary visual cortex here, and in V2, that's the other visual region, you see that this enzyme is much more abundantly present in these stripes. You have thick stripes, thin stripes, and interstripes is where the enzyme is less. Now, why this is so, and so why you have more activity of this enzyme here than there, or here than in the white regions, we don't really know, but that's just an anatomical feature again of primary visual cortex and V2, V1 and V2. So that's another functional, or well, primarily anatomical parcellation of visual cortex. Again, we're looking on top of visual cortex here. And of course, and then of course the other one is that these this orientation tuning, which is a very prominent feature of cells in primary visual cortex, is also organized in such a columnar fashion. And of course, that was explained in the video, but you can also, let me see, you can also see it here. And so if you present, in this case, a monkey with a vertical orientation, so a grating, which have all vertical orientations, and then look at the activity in visual cortex, in this case with optical imaging, it doesn't really matter all that much, you will see that, well, for this particular vertical orientation, you will see that all these black regions are activated by that. And then for another orientation, let's say here, this vertical, this horizontal orientation, you get other, maybe difficult to see, of course, but you get other regions which are activated. And if you would stick an electrode through the visual cortex vertically, you will find that all cells in such a black region respond to that same vertical orientation. And here, if you would stick an electrode vertically through the cortex here, you would see that all cells there in that column, again, respond to this horizontal orientation. So also this orientation selectivity is organized in such a columnar fashion. You have orientation columns. So you have all these different columns, you might say, ocular dominance columns, cytochrome oxidase columns, or blobs as they're called, orientation columns. And so that's explained here. You have all these different organizational components of primary visual cortex. Now, how can you combine all these? Because they're all present if you measure them independently. Well, this is what Jubel and Wiesel have called the hypercolumn, which is shown here. And so the idea is in one hypercolumn, and a hypercolumn is this. This is one hypercolumn. See? And in each of these, so there are 4 in total here. And the idea is that in each of these hypercolumns, in the center, you would have a cytochrome oxidase blob, which you have in each of these, 4 times in this case, 2 times, sorry. Of course, you have this organization of ocular dominance columns. So this is one ocular dominance column. This is the other ocular dominance column. Here again, left, right, right, left. And then, of course, you have these different orientation columns. So this is for vertical orientations. This is for quasi minus 30 degrees orientations. This is for horizontal orientations. So in this direction, you go through all the orientations. And of course, that happens for both the left and the right ocular dominance column. So this is sort of a theoretical, because of course it's important to stress that, there's a sort of a theoretical idea how you might combine all these different organizational features of primary visual cortex. But the other important idea of a hypercolumn is that one such a hypercolumn then is sort of a basic processing unit in primary visual cortex, because, in this processing unit, all the orientations are processed or detected. You have this cytochrome oxidase blob, which is, well, that's not dealt with in these lectures, but later on you may see that this is particularly important for the processing of color. Anyhow, and you have the two eyes being in such a column. So this one column contains all the information, if you would like to say so, contains all the information that is present at that particular location in the visual field, or that you would like to know about in that location of the visual field.
So to recap, you have like color information, orientation information, motion information, and both eyes in each hypercolumn.
Exactly. So, and then for each location in the visual field, you have a hypercolumn. So this is for, say, a particular location in the visual field. This is for the location in the visual field a bit next to that. This is for the location in the visual field a bit next to that. So you have like a few thousand of these hypercolumns in primary visual cortex, which are covering the whole the whole visual field. Only 4 of these are shown here, of course, but you have to imagine that you have, if you look at the whole of visual cortex, you would have, I guess, a few thousand of these hypercolumns, maybe even more. So that's the idea of the hypercolumn and the columnar organization of these different, orientations, ocular dominance, and cytochrome oxidase blobs. All right. I hope that answers your question, Noah. Maybe.
Yes, thank you.
Okay, thanks.
Great.
Welcome.
Good. I also edited the short answer on the discussion board. So we're going to continue with, I think the next question is about the bold response.
Yeah. Okay. So in the book, now for one thing, of course, you don't need to know what's in the book, but let me answer the question anyhow. When the stimulus is repeated, the bold response is lower in the second presentation compared to the first. Now, this is called repetition suppression. For one thing, the more important thing to know about this is that it tells you that if you get repetition suppression, that somehow this stimulus has been processed by this part of the brain, because otherwise, of course, if that would not have been the case, why would you find the suppression? Now, of course, the other thing is, where is this suppression really coming from? Now, there are different ideas, but essentially, it's not very different from, say, maybe you've seen this. If you have a snail, you touch his he has these 2 sensation horns. If you touch the horn once, of course, it will retract the horn, and and then it comes up again, and if you touch it again, it will retract a little bit less, because it gets habituated to this response. That's also a variety of repetition suppression, and it's something you will often find in any neural system. If you repeat the same stimulus, then every time you repeat it, the response will get weaker. It's very much the same that when you hear an alarm go off the first time, of course you give a big response, but if the alarm goes off like every 30 minutes, then at some point you will not respond anymore. And now, of course, here they say that maybe an explanation is that the neural response to the stimulus is more efficient and perhaps faster. Yeah, well, that's a more cognitive explanation that I don't really buy into that very much. But anyhow, it's a sign of that the stimulus has been processed, and if it's processed, then again, the response is weaker. Maybe because it's more efficient, maybe because it's just habituation. Anyhow, that's what it tells you, and that's how it's used in this part of the book. It's taken as a sign that, okay, you get a repetition suppression, so apparently this stimulus has been processed in that region, and that is then used to infer something about whether this region is selective for a particular stimulus. That's basically all this is about. Is that
clear?
Okay.
Okay, good.
Next question by Estee. Vision part 2, slide 11. I think that's another slide, but any other question is clear. From what you say, it sounds like the retinal corneal blindness is also called achromatopsia. That's not true. If I accidentally said that during the lecture, that's a mistake. Retinal color blindness is just what we call retinal color blindness, or color blindness for short. Achromatopsia is really reserved for this very different condition where you have brain damage, which causes you to not see colors anymore. Major differences between the two is that, of course, well, as the word says, retinal color blindness is something retinal, and cortical color blindness or achromatopsia is something which originates in the visual cortex. But they also have very different etiologies, because retinal color blindness is caused by this genetically determined absence of a particular cone type. And an important thing is this doesn't make you see everything in black and white. You just typically have difficulties distinguishing certain colors, such as, for example, green versus orange, which is a very common variety. But of course, that depends on the cone you miss. And this is indeed what is tested with these Ishihara plates. And so if you miss one of these cones, you will have difficulty seeing that it's an 8, and you will see a 3 instead. There is also a condition where you miss all cones, which is very rare, but then you indeed see no color at all. But a much more serious problem is that you don't see, that you have very unsharp vision, because you also need your cones to see sharply. If you would have to rely only on rods, your vision will be very blurry, so you cannot see many details. So that's a very different condition and very much more serious. And as I said, achromatopsia is a term which is reserved for color blindness, which is due to a cortical lesion. And this makes you see things pretty much in black and white. Also, of course, depends. You have different types and depends on the size of the lesion and things like that. But this is much more robbing you of the sensation of color, or at least that's the classic.
The conscious sensation, right?
Yeah, that also.
Because there's some evidence that people can still use color information on an unconscious level.
Yeah, that's of course a whole different. Yeah, that's true. You know, that's what you will also find with very many other cortical lesions, that you may see dissociations between, how you measure the deficit when you just ask people, or when you do a priming task or some other eye movement task, things like that. Then you may see all sorts of different outcomes. And so most well-known example, of course, is blind sight, that people have with a lesion to primary visual cortex, they don't see anything at all anymore. They don't have any conscious sensation, or at least that's the sort of classical take on it. But they can still guess, and they can still make eye movements, and they can still point to objects and stuff like that. So, but achromatopsia indeed is the loss of color vision, also color discrimination, due to a cortical lesion.
Good. Any more questions? You can unmute yourself or type in the chat, and in the chat I will repeat. Okay, good. Just waiting for 1 second. I think the next question was from Alessia. For the exam, do you have to know and understand the workings of magnocellular and parvocellular cells and their pathways? And Victor responded, no, you don't, except for what is on slides Vision 1, in the slides of Vision 1 on the slide 21 and 22. So that has been answered, I think.
Yeah, the answer is there. Basic point is that you have to know that they exist, and that they constitute different pathways that end up in different parts of the brain, constituting the so-called dorsal and ventral streams, which is talked on later on in Vision 4, slide 5, for example. But you don't have to know any physiological difference between them, so that's for later. So you don't have to know that the one is high spatial frequency, the other one is spatial frequency, stuff like that. You don't have to know that.
Great. I think I get a question here. Are cones and rods processed in different parts of the LGN? And that's question one.
No, they're not. Both cones and rods project. Well, of course, cones and rods don't project directly to the LGN. In the end, they all have to first feed into retinal ganglion cells. So let me share the right. Is my screen still shared?
Yeah, I think, but I see orthogonal organization components.
Okay, so if we go back to say this slide, of course, rods and cones, which you see here, all have to go through these bipolar cells and then reach the ganglion cells, and the ganglion cells project to the LGN. Of course, there's a difference between ganglion cells in the sense that you have these X and Y type ganglion cells or midget and parasol cells, but as I said, that's not something you have to know now. And these different types of ganglion cells receive different types of input from rods and cones, but that's way too much detail for now. Basic point is that both rods and cones in the end have to go through the information from both rods and cones have to go through the retinal ganglion cells, and these project to the LGN, and then to the brain. So it's not that each of these have, say, a different way of reaching the brain. They both have to go through retinal ganglion cells.
Good. And a related question: are rods giving information to the midget cells?
Yeah, that's this topic about midget cells and parasol cells, which has been left out of the lectures this year. So of course I'm going to give a whole lecture about that, but that's not. I don't think that's the goal of today. It's not a topic of study for this exam or for this year. So you don't have to know about midget cells and parasol cells.
But do rods project to the midget cells as well?
Yes, they do. In fact, rods and cones project pretty much in the same way to the LGN cells. The only thing being that the rods do this via a different pathway, different types of bipolar cells, also and they also have different ways in which they react. It's a fairly complicated story, which I cannot explain now. Just in I would have to show you some images because otherwise it gets very difficult. So, but yeah, rods also project to midgets and to parasol cells.
Thanks. I think I got a question from Bart. Does achromatopsia result only from bilateral lesions or also unilateral?
Well, the example in the book, and also, let me see, where's the, no, that's a different slide. I have to stop this sharing, do a new one. So for example here, this is slide 6. This is a patient with a focal unilateral lesion to V4 and a bit of other adjacent regions. And there you see that the deficit, and so how much different, what's here on the x-axis is how much different the two colors have to be for the subject to see the difference. So, for example, here in these parts of the visual field, so lower left, upper right, lower right, the patient can discriminate between this red and this, say, sort of orange. But it needs to be all the way up here to yellow for this upper left visual quadrant. So this is a this is a type of achromatopsia where the lesion is restricted to a particular part of the visual field, in this case the upper left quadrant. And the other parts of the visual field are sort of normal. They're not perfect, of course, but they're much less affected. That's over here the same thing. The patient can distinguish this green from, say, this sort of green bluish in these parts of the visual field, but it has to be all the way up to purple for the patient to see the difference between this green and this purple. So no, it doesn't have to be bilateral. Of course, it has to be bilateral for you to see the whole scene colorless, which is the classical achromatopsia patient, which is sort of illustrated. You may say here, of course, he doesn't see the orange, but that's just, you know, to indicate what the world looks like if you see everything without color, which is quite depressing. But that's no, so it doesn't have to be bilateral. That's by the way somewhat different from this akinetopsia. This is much rarer because it seems to be that it has to be bilateral for the patients really not seeing any motion anymore. Okay, but that's just a side note.
Excellent. So I get some more questions here in the chat. I got some from Maria. Could you please explain slide 15 from lecture 3? Maria, it might be good if you point out which slide deck it is exactly by clarifying the topic. You can do so in the chat or by unmuting yourself.
That's this one, I guess. Okay. So
That's correct, Maria. You can just say yes.
Yes. Can you hear me now?
Yeah, I can hear you now.
Okay. So it's the slide about defects in visual fields of left eye and right eye. It's from lecture 3 about vision, and it's slide number 15.
oh, it's lecture 3, so vision 1.
Yes.
Ah, okay, that's a different one.
Thanks.
You pointed out something on this slide, but
wait a second. Wait a second, so that I can get to the slide. Can you see it? Can you see me scrolling?
Yes,
through?
yes, I can.
Okay, so which slide? 15.
15.
Ah,
yes,
yeah.
this one. You pointed on the slide, but we couldn't see it.
Yeah. Okay. Okay. So yeah, this is also something that was asked via mail, but was that also by you? But anyhow.
No.
Oh, okay. So anyhow, the basic thing is, of course, that you have to know this projection of the two visual fields to each of the two hemispheres, so that you have the left visual field projecting to this right side of the left eye and the right side of the right eye. Then this part crosses over in the optic chiasm, and then this one stays at the unilaterally, and then they both project to the LGN, and then from the LGN you get to primary visual cortex, so that in the end the left visual field projects to the right visual cortex. So as I say in the lecture, also a mistake that's often made is that people think that the left eye projects to the right visual cortex. That's wrong. It's the left visual field that projects to the right visual cortex. And of course the other way around, right visual field projects to left visual cortex. Now what's shown in this slide is the different lesions that may occur and then different field defects that you will then get. And so if there is a lesion sitting here, cutting your optic nerve, or maybe if you just lose your eye, which is a more common cause, I would say, of course you would have a visual field defect that looks like this. So you see the whole visual field in the left eye and you see nothing in the right eye. That's fairly logical, I would say. I will go into 2 in a minute because that's a bit more complicated, but let's go through 3 first. 3 is when you have a lesion after the optic chiasm. And so after you get this crossing over and stuff that I just explained. So you have it sitting there. Now, of course, then if you can just follow the yellow lines, of course. So then what you would lose, of course, is that in both eyes, you no longer see the left visual field. Yeah, because maybe better explained here. So if you would cut these, if you would cut the optic tract, as it's now called here, you would lose all information from both eyes in the left visual field, which is shown here in red. And that's indeed what you see here. So now you get in both eyes that you miss the left half of the visual field. So it doesn't matter whether you close one eye or not, you will still miss the left visual field. And of course, if the lesion would be here on the left side, you would get the opposite, both eyes' right visual field would be damaged. Now, what you get here with 4, 5, and 6, that's actually a bit confusing because it just looks like they are selectively deleting the part of the one eye, but that's not what you take from this. This is that if in this optic radiation, it's called, you have upper and lower parts, and depending on whether you lesion the upper or the lower part of the optic radiation, you get a deficit in this case, of course, upper left visual field or lower left visual field. Because it's not only the case that left visual field projects to right visual cortex, but also lower half of the visual field projects to the upper part of your visual cortex, and the upper part of your visual field projects to the lower half of your visual cortex. So that's what's illustrated here. So now you damage just let's say half of the optic radiation, and then you get a damage to the, of course, half of the left visual field. In this case, the upper half, or in this case, the lower half. Now, what you see here, this small circle or a quarter circle, and what you also get here with this half circle. So if you would have a complete left visual field lesion, that's something, but it's a bit controversial, but that's something that has allegedly been shown, that if you have damage to visual cortex, so that's why the lesions now sit here, that you don't get a full loss of the left visual field, but you get what's called macular sparing. So a sparing of the macular region or the foveal region, allegedly because this region is represented both in the left and in the right hemisphere simultaneously. But that's a bit controversial whether that's really the case. But either way, this macular sparing and foveal sparing, those are details that you don't really have to worry about. The main thing that I really, really, really would like you to know and understand is that the left visual field projects to the right visual cortex and not the left eye projecting to the right visual cortex. Because every year we always see even third year students which still get this wrong. So if you understand that, I'm perfectly happy. Now let me go to the final one, which is this second one. It's a very special case where you have left visual field deficit in the left eye, right visual deficit in the right eye. That would happen if you have a lesion here at the optic chiasm. So for example, if you cut the optic chiasm here, you can just follow the projections for yourself. But then of course what you get is that in this left eye you still have this. So you still have the right visual field, which is indeed what you get here. And in the right eye you still have this projection, and the other one is not going through, and you don't have input from the contralateral eye here. So you have right, you still have the left visual field in that eye, and a lesion to the right visual field. Now you might wonder, well, how the hell is that going to happen? That you have a lesion which cuts this optic chiasm exactly in half. Well, of course, it doesn't really ever happen like that. Well, unless you have a very strange accident. But it can happen if you have a tumor of the pituitary gland, because the pituitary gland sits right at that location where the optic chiasm sits. There you also have your pituitary gland, which produces all these hormones. You may know this from high school. If you have a tumor there, this tumor may grow, of course, and then sort of shred apart, tear apart the optic chiasm, so that you get sort of a lesion like this. So that could be, if you have a lesion like that, that could be one of the first symptoms of you having a pituitary gland tumor. Well, maybe not the first symptom, but it could be a symptom of a thing like that. Okay.
Okay, thank you.
Okay. Anything else?
Yes, sure. I think the next question is, what's the difference between a postsynaptic potential and an action potential?
Ah, okay. That's a fairly fundamental difference. Let me share that thing then. Now the action potential, well, no, let me start with the postsynaptic potential. Where is it? What is that? Postsynaptic potential sort of starts with the release of neurotransmitter in the synapse, which you can see here. This neurotransmitter, which are these tiny blobs, are of course released by what's called a presynaptic axon. So this is the axon that contacts another cell, and this is then the other cell. This is called the postsynaptic neuron. This is called the presynaptic neuron. This presynaptic neuron releases these chemicals, this neurotransmitter. And typically what happens is that this neurotransmitter will then bind to a receptor which is sitting there. And this receptor typically is a channel, which then opens. Maybe, by the way, also closed, but it mostly opens. So that now, for example, sodium ions can flow in. Of course, there can be also different channels, for example, chlorine channels or anything else, but let's take sodium as an example. So now sodium flows in. And then if you go back to this slide, so here you have this channel opening. So here is the neurotransmitter, which is bound to the channel, then sodium flows in. And then you get a depolarization, which you can measure here. So depolarization of the membrane potential. Because it's positive ions flowing in, it gets less negative. And that's what's called the excitatory postsynaptic potential. Now, if it's just one synapse, of course, it will be a very tiny excitatory postsynaptic potential. But if it's more, you know, these EPSPs then add up, which is what is shown here. So then these EPSPs will add up. So this is a single EPSP, and then after a while another one. But if you have two at the same time, they will add up. So here's one, and then immediately afterwards there's another one. And at some point, of course, they will reach this threshold where the voltage-gated sodium channels open. And then you get this, well, it's better illustrated here. And so excitatory EPSP here, another EPSP here, and then you reach the threshold. And then these voltage-gated channels will open, which will then cause more sodium to flow in, and then more sodium-gated, voltage-gated channels will open, more sodium will flow in, so that you get this very rapid depolarization, which we call the action potential. Sort of an all or nothing process.
Yeah, and one thing I would like to add here, that postsynaptic potentials is basically the input into a neuron, whereas the action potential is the output of a neuron. And I think this is a crucial thing to state, because if we're talking about EEG, for example, we're not picking up on the action potentials itself, but we're picking up on the postsynaptic potentials, and then pulled over a large number of neurons.
Yeah, that's another.
So in that sense, I think that's quite important distinction, because in humans with EEG, with fMRI, we like to. So this is never linked to the action potential itself, but mostly to the postsynaptic potentials that you measure. Maybe Victor wants to comment on that as well. But
yeah, well, that's basically what I explained in this recording neural activity thing. So maybe we can share that for a second. Yeah, so if we, so where's that explained best? Maybe not exactly, but these inputs causing these membrane potential changes. So these EPSPs or IPSPs, by the way, they generate these electrical fields in and outside of the neuron, which then, if all these dendrites have the same orientation, as you will see in cortex, for example, then all these tiny, of course, they're very tiny electrical fields may add up to something which creates a larger electrical field that you can even measure either on the cortical surface or maybe even on the outside of the head with an electrode on the skull or on your scalp. So it's not only the case that EEG is only about these synaptic potentials, but it's even worse. It's only about synaptic potentials which are generated in these apical dendrites in the superficial layers of the cortex. Of course, there may be some other contributions, but for example, what is that showing? These if you look at action potentials, of course, action potentials will also generate these electrical fields, but axons typically run in all sorts of directions so that all these electrical fields will cancel, which is the same what you also get with dendrites if they are arranged like shown here. So here you have, for example, electrical field going from here to here, another electrical field coming from there to there, and if you then would look at this electrical field from the outside, they will all cancel to nothing or something that you cannot measure. So that's a very typical problem of EEG. Because you measure at a distance, you have, well, actually most of the electrical signals of the brain canceling out each other so that you don't record them at all. And only if you're very lucky, so like shown here, that you have these, for example, apical dendrites, which are all pointing in the same direction, so generating electrical fields all in the same direction, then you can actually record something from the outside.
So it has to do with the neurons being very asymmetric in that sense. If the neurons are very symmetric, you cannot measure them as well.
Yeah.
Great. Okay, I think we're about to close up for Victor's part, and I'm going to continue for an additional hour. We planned in a new meeting tomorrow at 10. So if you have any more questions, feel free to post them. Then we host this session again before the exam. Of course, it's important to also state that the trial exam is a very representative. So the weekly assignment contains very representative questions. It's only 13. So if you do them twice, you get the same questions. So that's something that is important to state. I'm going to close off with, I think, the final question to Victor. I'm sorry if I missed any of this. I'm multitasking. That's never good to do.
The final question is about TMS and TES.
No, but I'm going to treat that in the next part because I discussed that in quite some detail. So the question is why the sarin gas is an example of acetylcholinesterase inhibitors, the PowerPoint page 41 of membrane potentials. So why is that?
Okay. That's the lecture I'm still sharing. Can you see me scrolling through it?
I can see the EEG thing, depending on the location of activity.
Oh, then I have to stop. Then I have to reshare. Okay, sorry. Go there. Okay. What is this bit? Yeah, this is sarin gas. Now, as I just explained, what neurotransmitters do is, they are released in the synaptic cleft, shown here, and then they bind to the postsynaptic these postsynaptic channels, so that these channels can open, and then you get the EPSP, so that the neuron gets activated. But what happens after that is that this neurotransmitter is then released again from the channel, because otherwise, of course, it would stay there forever, and you would the neuron would the channel would be open forever, and you would get sodium flowing in forever, and you would get continuous depolarization. So it's typically very rapidly taken up again, and there are different ways different things can then happen with this neurotransmitter, which is shown here. So, for example, it can be reuptaken. Is that a word? There's reuptake by the presynaptic neuron. So there's a specific channel which takes up the neurotransmitter. So that's, again, it is inside the presynaptic neuron. It can be degraded enzymatically. So there's an enzyme which breaks apart the neurotransmitter, so that's no longer doing its job. Or it can just diffuse away from the synapse. That's, of course, a very weak mechanism. But anyhow, these are all very important to prevent continuous opening of this channel and continuous depolarization. Now, if we look at this enzymatic degradation, there's one very important enzyme which does this for the neurotransmitter acetylcholine. Acetylcholine is a neurotransmitter that's very often used in muscle transmission, so the transmission from a neural signal to your muscles, but also in the brain. But it's most well known for the transmission of the signal from a neuron to a muscle, which works the same, also with the synapse. So acetylcholine binds to this cholinergic receptor, which causes the channels to open, etc., etc. But what then happens is that at this postsynaptic cells, you have an enzyme which is called acetylcholinesterase, which breaks apart the acetylcholine into acetate and choline. And this is then taken up by the cell again, and then can be used again to make new acetylcholine. Now, if you would inhibit this enzyme, acetylcholine would no longer be breaking apart, so it would continue to work on this receptor. So this receptor would remain open. You would get continuous depolarization, etc. Now, indeed, there are such inhibitors, acetylcholinesterase inhibitors, such as many venoms. When you get bitten by a snake, sometimes the toxin contains acetylcholinesterase, but there are also other ones. And sort of sadly well-known example is a nerve gas which is called sarin, which was used in this Tokyo subway attack, but also in other, ugly wars, you might say. And of course, what this does is inhibit the acetylcholinesterase, so that the acetylcholine stays bound to the postsynaptic cell. Channel stays open, so the muscles keep getting activated, or the neurons with the postsynaptic neurons, but in this case, the muscles keep getting activated, so you get spasms, muscle spasms. Continuous activation of your muscle is what we call a muscle spasm. And of course, if you have a muscle spasm, for example, that's very problematic if you want to breathe, because in breathing you have to, of course, use your muscles and then relax them again, use them and relax them again. That's what breathing is. And then if you're constantly like this in a continuous state of breathing in, of course you die of asphyxiation. And that's what happens. It's a very horrible way to die, of course. That's what happens when you are subject to these kinds of acetylcholinesterase inhibitors.
Right. Thanks. This is everything for now for you, Victor. I'm going to continue for some more time. And again, let me restate that we're going to continue tomorrow at 10. So if you have any additional questions, please do so. Victor will be around between 10 and 11. Then I'm taking over between 11 and 12. And I think if you also did the weekly assignment, this is a very good indication of where you basically mastered all the material. So let me say thanks to Victor, and see you again tomorrow.
Okay, bye-bye. See you.
Okay. Great. I'm going to take over. So let me share. Let's see. I want to pin video. Okay, there. Good. I got some questions, and now I definitely need to multitask. One of the there were a couple of different questions on the board. I can answer some questions that I get via YouTube and via the chat. Please be patient with me because I have to collect all the materials here. So one of the things is I think I'm going to be talking about EEG and oscillations. The question was where I could repeat some of the information that was on the slide. So this takes a bit more time to explain everything. So in that sense, I think I will save that for a bit later. One of the other questions was about, so Noah, I'm coming back to your question later on. One of the questions by Marloes was, what are exactly columns? I think Victor was talking about this a bit. If you look across, for example, primary visual cortex, you can see that in one specific column, it only processes one specific orientation. If we go into the motor cortex and basically go with an electrode through all the different cell layers, then you can see that all the different cell layers maximally respond to one direction of movement you make. So it's basically a column means that all the cell layers that are involved in that specific part of cortex are all responding to the same feature or coding for the same kind of facial features to the same kind of cognitive aspects. So that I think it's a specialization across these different layers, these 6 different layers of cortex that are all the same. This was in the anatomy lecture. I hope Marloes is here. It's hard for me to. No, I don't think so. Please chat or unmute yourself in case you want to ask anything else about these columns. If it's clear, please say clear or something like that. Okay, Iris asked some questions about the ventral and dorsal root of the spinal nerve. Let me just share my screen. I think it's in the anatomy slide. Iris, could you please say which slide it is so it speeds up everything?
I don't really know which slide it is. I had it in my own summary.
Yeah.
Oops, sorry, I don't know what slide it is.
Can you now see anatomy of the nervous system?
Yes.
Okay. I think it's quite early on in the slides. Yeah, there we go. It's probably here.
Yeah.
Slide 12 of the anatomy class. What you basically see here in the somatic nervous system, basically carrying information to the motor system through the spinal nerve, and carrying back information from all the touch receptors in our skin back to the nervous system. And there is quite a clear separation here. So the ventral parts, ventral meaning the stomach parts, that is basically carrying all the motor signals from the spinal cord to the motor, to our muscles. Motor output basically are these ventral roots that you see in this picture. If it comes down to all the feelings or all the information we gather from the receptors in our skin and the receptors in our muscles, feeding back to the somatosensory cortex, that all is the dorsal part. So that's the back part. So there is a separation. If you would cut, say, the ventral roots, then you cannot move your muscles anymore, but you can still feel. If you would cut the dorsal part, then you could still activate your muscles, but you don't feel anything anymore. Is that clear?
Yes, thank you. And just for my own clear mind, the big column in the middle is the same as the spinal cord.
Yeah, that's basically one part of your spinal cord.
Yes, okay. And there's different spinal nerves on, there's like more than one, like 20 something or something, right?
Yeah, you're right.
Yeah, okay.
Well, that's a bit of more detail. I think the main thing here is that we have the separation of motor output, ventral, and the dorsal input, and that's the dorsal part.
Yes, okay,
that's the most important thing. So you see that these two different aspects are clearly separated.
Yes.
So I'm going to stop my share again because it's difficult to read and see the chat and respond to everything at the same time. So let me check on the other questions that we have. So columns is treated. I had some question by Yasmina about the difference between stellate and pyramidal neurons. That's also the anatomy class. So again, let me share my screen. That's desktop 2. I think here we can see the topics. Can you see the anatomy slides here? So still somatic nervous system. Please somebody unmute because I cannot see the chat at the same time. Yeah, good. I'm just going to move forward to the later parts of the lecture here. So here from slide 65, I was talking about the organization of the cortex in different layers. So here you see the 6 different layers of the neocortex, clearly separable. And again, if we're talking about, if we inspect carefully what we see in terms of the kinds of neurons that are most prominently present in the cortex, we see this typical pyramidal neuron, very asymmetrical shape of a pyramid. When we were talking about EEG, for example, this is typically the neuron that you can measure with EEG. The stellate neurons are quite symmetrical, so all these different electrical fields are canceling each other out, so you are unable to measure this using EEG. So if we're talking about, again, about EEG, we're measuring a summation of the postsynaptic potentials, and this is really related only to these very asymmetric neurons, these pyramidal cells. If we're talking about the basic difference between a pyramidal neuron and a stellate neuron, the pyramidal neurons are basically the big motors of our cortex, of our entire brain, and they only operate on glutamate, an excitatory neurotransmitter. So that's a very important thing to know. If I go to the stellate neurons, you can see them here. They're much smaller. They're star-shaped. They're symmetrical. And we have 2 types of stellate neurons. We have excitatory. They also work on glutamate. And we have the inhibitory stellate neurons that work on GABA. If you basically knock out the GABA neurons, the inhibitory neurons, then the cortex fires itself to death within minutes. So it seems to be some kind of fail-safe to keep the excitatory system in check. And that's also something related to, if we're talking about EEG, the main reason why we see these kinds of oscillations in the EEG is that if the pyramidal cells and the excitatory stellate neurons become active, they activate these inhibitory stellate cells, and they shut down the entire circuit. So we see increase in activity in pyramidal cells, an increase in activity in this inhibitory stellate cells, and that shuts down the neural activity. So a natural consequence of that would be an oscillation that would follow. So that's basically integrating a bit about the oscillations of the EEG. Why do we see oscillations? So this is, I think, the most important reason why we see oscillations. Activity goes up in these pyramidal cells, activating the inhibitory stellate cells, and then dampening activity again. The most important distinction here that you need to know is pyramidal cells are the big motors using glutamate. Stellate cells can serve different functions, and they come in two neurotransmitter flavors, either GABA, the inhibitory circuit, or excitatory, and then it again uses glutamate. So any additional questions on this? Please unmute yourself.
I had a question on the slide. You said that, or it says that pyramidal neurons, under it says projection neurons.
Mm-hmm.
Are those the same thing?
What you can see is that these pyramidal neurons here,
Yeah.
and especially the one in the middle, have a very large axon. So they can communicate across long distances. It can go to the spinal cord. It can go from one side of the brain to the other side of the brain. So they are suited to basically take care of communication across brain regions. And stellate cells are very local. So they basically operate even within a column, and sometimes a bit across columns, but specifically within one brain region.
Okay, thank you.
Any other questions? Please unmute yourself and speak, because I'm unable to read the chat and navigate and do everything at the same time. Okay. So this was basically the stellate neurons and what we should, okay, stellate pyramidal neurons. I answered the question about columns. I answered, I think, the TMS transcranial electrical stimulation and magnetic stimulation question is something that I want to treat. That will be quite fast, I think. The EEG question is a bit more difficult to answer, and then that takes a bit more time. Okay, so let me start off with the TMS and TES questions. Do you see methods of cognitive neuroscience?
Yes.
Good. I think I have to go to, say, slide 20 or something. 21. Loads. Yeah. So that's so I think I was talking about these different correlational techniques that we can use, and also these causal methods. The thing is, I think a very important distinction here is, if I would ask you to participate in one of my experiments, I stick electrodes to your scalp when using EEG, or put you in a MRI machine when I want to use a functional MRI. I basically repeatedly show you an image or ask you to think of something. So I have repeated perceptual, cognitive, and motor processes there. And if I repeat this often enough, then I can average and see some kind of brain signal that I can link to these different events that I've shown you during during the experiment. At the same time, you're probably quite bored during the experiment, thinking about what you're going to do next, what you're going to eat, etc. So all these other things are being measured in EEG and fMRI as well. If I repeat it often enough, then the mostly likely explanation that I see these brain signals is that they were produced by the experimental paradigm you were in, but it's still a correlation. This is my assumption. And I think that is one crucial thing to understand with all these brain measurements, that it's likely that it's linked together, but we're not 100% sure. If we're taking these causal methods, then we're actually changing brain activity and see what you can do better or what you can do worse when I zap you, for example, with magnetic stimulation. Please also unmute me here if you want to ask additional questions. I think the main difference here is when we're applying transcranial magnetic stimulation, we're mostly stimulating quite a small region of the cortex. And that's one big distinction with electrical stimulation. We don't know exactly which part of the brain we're stimulating. So TMS is much more precise than electrical stimulation. So if I know this part of the DLPFC, this specific 1 by 1 centimeter I want to stimulate in the DLPFC, for example, or anywhere else, then you should always use TMS, because in electrical stimulation, you zap the entire prefrontal cortex, so you don't know for sure which region you're targeting. If we're talking about TMS, it's listed here in red and green. So if you're applying TMS to a speech region, for example, while you're trying to speak, then it always intervenes with task performance. And this is a crucial thing. So TMS, zap, you either see nothing because the region is not involved in the task that you're doing, or you see a decrement in performance. If you do it before the task and then zap somebody for, say, 10 minutes, depending on the characteristics of the TMS, you can either get better after having applied TMS, or you can do worse. So you can basically twist the volume knob to both sides. But that's really first you TMS, for example, for 10 or 20 minutes, and then you have people perform the task. And then you're changing the baseline activity in the region that you targeted. If you have any questions on this, so this is one of the things that is important. TMS is much more specific in space, and you can, during the task, it always reduces performance, unless you're basically zapping the wrong region in the brain. If you first TMS and then see what happens afterwards, then it can go both ways. You can either perform better or perform worse. If we're talking about electrical stimulation, it's a way cheaper way of doing it. It's quite a safe way of doing it. And I think one of the things that if you are an elite athlete, for example, you cannot take a TMS machine with you quite easily in a backpack because it's way too heavy. So then you have to refer to electrical stimulation because you basically have no other way of doing this. So you have to weigh in, like, what do I want to be doing with my method? If you want to be very precise, very rapid, then you need to use TMS. If you want it to be very cheap, very safe, very lightweight, then you have to use electrical stimulation. And if you don't care where you're specific in what you're stimulating, then electrical stimulation would be a perfect solution. So any questions about this? It's basically weighing the cons and the pros against each other. And the same thing is important to do when talking about functional MRI and EEG. If you really care about where activity is located, then you have to use fMRI because you can use the millimeter precision there. EEG is more like centimeters precision. If you really care about time, then you should go for EEG and not so much fMRI.
Can you combine TES with fMRI?
Yes, you can. But typically it's quite challenging to do this. So you need lots of time. And actually the magnetic field of fMRI also generates currents in the wires of the electrical stimulation device. So you have to insulate it very carefully. So it's difficult, but it is possible. Okay. Any other questions about this? It's basically weighing in the pros and the cons, and that's something that I think it's important to make an educated decision. Yeah.
Sorry, is there any other way to have an experiment to have a causality besides TMS and TES?
Yes, in this entire lecture, we had lots of we discussed quite a lot of different methods, like electrical stimulation, like magnetic stimulation, and these are typically the two things that are quite easy to use in experiments with, say, first-year psychology students. Of course, there's also other ways like optogenetics. Then you basically infect the brain with rabies virus, and then it responds to different kinds of lights. You can imagine that if we try to do this in humans, that this won't go through the ethics committee. So this is something that we can use in very restricted test environments in animals. And I think that also goes for this intracranial stimulation. So in these cases, in some cases, if a person has a severe epilepsy, that might endanger them. And then you actually want to open up the skull, insert electrodes directly on the brain to pinpoint where the epileptic seizure is starting. And then you can also do microstimulation using these electrodes. But these are very rare cases, I would say. And you basically need to base your experiment on a patient that is available to you. Pharmacological experiments, that's something that you definitely can do. We have done some experiments where we, for example, use NMDA antagonists like what was it again? I have to remember this. It's a colleague of mine. It will come to me in a bit, but you can typically administer specific substances that change the neuromodulator system. And then I think ketamine, that's something that has been used for ages when you try to put somebody under narcosis. It's a party drug nowadays, but it basically operates on the NMDA receptors. And you could do consciousness research doing that. The problem of these things is that it kicks into the entire brain, so it's not specific to any region. Experimental lesions are something that you could do with specific animals. And I think the entire field of the causal method started off with naturally occurring human lesions that happen all the time. Specifically around the temporal parietal junction, it's quite common that people get a bleeding there. And then you get all kinds of problems in for example, speech, in, for example, attentional functions. But I would say, like, practically speaking, if you in the future would want to do some kind of causal experimentation, then TMS and TES are the only methods that are in common use. Sorry for the long answer.
Thank you.
Good. So that's that. I think the EEG question is something that is important to be talking about next. For that, I'm going to start up let me just stop sharing and see where there's anything. Okay. Okay, I get some questions here. I will look at the questions later. Let me go for the EEG part of the lecture. I think it starts at slide 20 or something. No, 30. Okay. Yes. I think this is important to know. We always ask exam questions about these things. If you have any questions, unmute yourself and let me know where anything is unclear. I'm going to share the screen again. Yes. Okay. And I'm basically starting at slide 29 here of the chapter 3 lecture on cognitive neuroscience. What do you measure with EEG and MEG? Okay. The complex story, is that visible for everybody?
Yes.
Yeah.
Good.
Yes.
So this basically links back a bit to what Victor was talking about. In Victor's lecture, he talked a lot about action potentials. He talked a bit about postsynaptic potentials. And I think this is one crucial thing to remember here. If we're talking about EEG and what we measure with EEG, we're talking about the postsynaptic potentials. So it's about the input to pyramidal neurons that we're talking about. So once an action potential has been generated by the presynaptic neuron, it basically releases neurotransmitters to the next neuron, the postsynaptic neuron, and there it induces postsynaptic potentials. So that's one of the important things. One of the other things is that if a large number of neurons at the same time produces these graded potentials or these postsynaptic potentials, they might add up to be detected far away. And that's something that you see over here. So I don't want to sound esoteric here, but if we think about an aura or an electrical aura, if all these pyramidal neurons or electrical systems in our body line up and are asymmetric enough, then you will be able to measure that outside of the body. And that's exactly what happens here. So only if we're talking about these pyramidal neurons that are very asymmetric and all the dendrites here line up and point into the same direction, then we can measure this outside of the body. So if we add up all these different graded potentials across different neurons, they will form these so-called local field potentials. Again, let me stress this again. Local field potentials reflect the combined input at the dendrites. So it's never about action potentials. Okay?
Can you explain if these fields can be correlated with some type of neural activity, or how does it relate to the overall cognitive functions of humans?
I think this is a bit difficult to answer, but say I'm imagine that I have one EEG electrode directly sitting on top of primary visual cortex, right? If I suddenly observe a signal in that electrode, that would mean that at that moment in time, activity arrives. So these axons from the LGN basically provide input into primary visual cortex. Okay.
Okay, just one, and you mentioned that these are not related to action potentials, but no. There is a correlation between the action potentials and these fields, right? So they always happen.
If there's no action potential arriving, then these postsynaptic potentials won't happen. So the action potentials will produce postsynaptic potential, but they all have to be the same polarity. So if they're all positive, or if they are all negative and add up across a large number of these pyramidal cells, then you will start measuring it with EEG. If it's exactly the same amount of inhibitory and excitatory inputs, then it will cancel it out in the electrical field, and you won't be able to measure that with EEG.
Okay, so then EEG measures also neural activity that it's not really translated to action potentials.
Exactly. So, and that actually, if you observe action potentials, they don't always lead in the postsynaptic neuron to a new action potential. So it can just be gone. So in that sense, this is a difficult translation to make. You do see something in the EEG, and it might not lead to a new action potential going to a different brain region, for example. So if you can link different parts in the EEG to behavior, to different perceptual stages, then yes, then you can correlate that. So it's immediately evident then if the EEG signal not always produces a new action potential in the neural tissue, that it is only correlational.
Okay, thank you.
So this is one clear thing that you need to remember. If we're talking about the EEG, here you see the open field example. So all these pyramidal cells are very nicely aligned next to each other. And then you can start measuring this outside of the body. In a closed field, for example, in all these subcortical structures in the brain, it's much more disorganized, and the neurons are not neatly organized in different layers as neatly organized in layers as in the cortex. So still, if you put in a depth electrode, you can measure the activity, but all the orientations point into a different direction. So the net sum here is 0. So you won't be able to measure that outside of the field. So you're not able to measure that far away from where actually the neural activation is happening. Okay. So that's basically postsynaptic potentials that add up across like a large number of pyramidal cells that all point in the same direction. That's what you're measuring, neurally speaking. So never fall for the trick question that it's about action potentials here. So then something that I was talking about before, like the difference between these pyramidal cells and these stellate cells, these interneurons, these stellate interneurons. So this, if we're talking about oscillations, the main reason that we see oscillations in the EEG is the phenomenon that when a pyramidal cell, an excitatory neuron, becomes active, they will start activating inhibitory neurons. These inhibitory neurons will then put a brake on the excitatory neurons and will afterwards silence themselves. So they are auto inhibitors in that sense as well. And that leaves room for the for the excitatory neurons, these pyramidal neurons, to become active again. So this is the basic reason why you always see these oscillations to start off with, because these excitatory neurons are coupled to inhibitory neurons. Is this clear?
Yes.
Okay. Of course, there's also other reasons. So if we're talking about, this first part that you see here in reason 1, that's happening everywhere in the neural system. So this is the basic reason why if you stick an electrode anywhere in the brain, you will start to see oscillation. If we're talking about other things, so in the brainstem or in the thalamus, it can gate information and make the entire visual cortex more active or less active. And these pacemaker cells, they basically set the rhythm for activity in, for example, the visual cortex. And then you can see the entire visual cortex responding to this drive from a remote region, and produce activity in lots of cells at the same time. So this is a second region. And this is not always what we can see when we look from the outside. And that's something that, like, what do we want to take from this communication? And that's if different brain regions start communicating with each other, we can observe that in different frequency bands. So the higher the frequency, say we see something like 80 hertz, then it's typically very local brain communication, very specialized regions of the brain doing their normal job. If we see very low frequencies, then lots of brain regions are doing the same. And that basically means if your entire visual cortex shows a very low frequency, then all these neurons, all having different specializations, are more or less in sync with each other, telling you that they're not really doing anything at this moment in time. So global communication, but also inactivity. And finally, I think a very boring reason, heart and heartbeat related, respiratory system related. If you exhale, then you have less oxygen and you see a dip in oscillations as well. So these are the boring reasons that you see back in the oscillations as well. Okay, I'm pausing for just 1 second. Everybody following me up to this point?
Yes.
Just one question. So the higher frequency, is there a difference between the higher frequency and lower frequency, let's say in relation to the default network of the brain?
I will come to that. Let me skip ahead here. So if we're basically talking about EEG and what you observe, in this slide 43, you can see that if you observe very high frequencies, so 30 to 100 hertz, then typically the brain is very active, or the brain region where you observe this is very active. So if you see gamma frequencies over visual cortex, you know that visual cortex is really getting a lot of input at that moment. If you see very slow oscillations, so very low frequencies, then typically all the neurons are doing the same. So there's no specialization. So it's basically in a rest state. And that's typically also what you observe when you're looking at the EEG, for example, when somebody is sleeping. If you see high frequencies, low amplitude, you would say somebody is awake, excited, etc. When you see very slow oscillations and with a large amplitude, then you would conclude that somebody is asleep, or if the frequency shifts down, so going from say 4 hertz to 2 hertz, you would conclude that somebody transitions into a deeper sleep phase. Does that answer your question?
Yes, thank you.
So that's, I think, one of the most important things. So we're talking about what do you measure with the EEG? Postsynaptic potentials average across a large number of pyramidal cells. I'm just repeating here. The oscillations, where do they come from? Well, this is the basic reason. That's the way that these pyramidal neurons and interneurons are wired. In addition, there are pacemaker cells, and when brain regions communicate with each other, we also see this surface in the oscillations that are present. If we're talking about what can we do with EEG signals, so all the things that we're measuring in the EEG, if we're starting measuring this at the electrode, we see all different sine waves that differ in amplitude, frequency, and phase. And in this example here, do mind here that I think the bottom right figure is wrong because it's still amplitude 1. You have different signals. Say these signals originate from different regions in the brain, and you measure a mixture of these signals on the electrode. So a combination of these 4 waves would give you something that looks like this. And this is just mathematical summation of all these postsynaptic of these local field potentials that you measure at the same time at the electrode. You don't exactly know where the signal comes from. You don't know which amplitude and frequency is linked to which source in the brain. So you basically have to do a couple of tricks to use this information. And this would be a more typical kind of thing. If you have like 10 different signals, then it adds up to a signal that you can see in the top of this graph. Of course, there's noise involved. Here, there's more noise. Here, there's even more noise. At a certain point, you cannot see the signal anymore because there's too much noise in the. There's too much noise that you measure. But what we typically want to say, like if we assess the frequency of the signal, you can say something about active or inactive. You can say something about local communication and global communication. And this is basically what this slide 44 is depicting. If it's high activity, local activity, and very active. If it's low frequencies, then either it's global brain communication or you're deep asleep. Questions on this?
No, clear.
Okay. Then I'm finally going to wrap up on how you can use EEG. So one of the things that is quite a miracle to me, I think, here we can see a visual stimulus arise in the left visual field. This projects to the right visual cortex. So here, this is time locked at 0. We can see nothing happening yet. Then 25 milliseconds later, nothing happening yet. And then in the fourth part, 75 to 100 milliseconds, then we suddenly see a positive dipole happening. So there, the information starts to arrive in primary visual cortex. Then the signal is sent forward to the parietal cortex that you can see happening in 125 to 150, more prominent in 150 to 175. And then at 225 to 250 milliseconds, the information is sent back to primary visual cortex again. So you can see the evolution of information coming in at a certain point, going to a different region, and then coming back to the region it originated from. So you can basically track which brain regions communicate with each other in which order across the entire brain. Again, if you are interested in the event-related potential, so you show a flash of light to a person, you do that over and over and over and over again, you get these, you get rid of the noise, and these different peaks start to surface in the averaged EEG. Positive waves are termed P. So the P1 here is the positive potential that appears about 100 milliseconds after you've shown a stimulus. The P3 is typically 300 milliseconds after you've shown a stimulus. And these are all linked to specific neural events. So for example, the P3 happens when you upload something into working memory, then typically you see a P300. The N2 is typically what you observe after a visual percept is fully evaluated. So we can label these different peaks like, okay, something happened here. I see a P3, something was uploaded into working memory. Okay? So we can basically link this together to specific discrete neural events. And that's something that is important to know if we're talking about event-related potentials here. We can couple these by years of research to different information processing steps. Also, if, for example, person A has a P1 that happens at 90 milliseconds after stimulus onset, and somebody else consistently we see it happening at 120 milliseconds after stimulus onset, then we can conclude that the brain of person A, when seeing information, operates faster than of person B. Okay?
Yes, that's very clear. Thank you.
And finally, I think this is a much more sophisticated technique that you could use. We know that all these sensors that we measure with the EEG electrodes, the actual sources of the information, they have different amplitude, different frequencies, and we can also tease this apart. So we can here inspect how strong is each frequency present in the signal at which moment in time. And this is a very sophisticated technique where you need to have a very strong computer, lots of processing power to tease this apart. And ERPs is actually quite simple to do that on a 10-year-old computer. And then you can look at these different frequencies. And typically what you do is if you, for example, see brain region A communicating, so at a certain moment in time, say I'm just pointing here, between 150 and 450 milliseconds, we see, say, a 12 hertz burst. And we see that same 12 hertz burst in another brain region, for example, in prefrontal cortex, more or less at the same time. Then we start concluding that because the oscillations that we observe here, because the frequency that we observe here happens more or less at the same time in two different brain regions, that these brain regions are communicating with each other. So this is mostly used to see which brain regions are communicating with each other. And if in this case, for example, brain region A starts slightly earlier than brain region B, then we can infer that brain region A is communicating towards brain region B. Is this also clear?
Yes, thank you.
So this is mostly on the conceptual level. We want to know which frequencies are present in the EEG at what specific time to conclude something about direction of communication between these brain regions. Because if we're just looking at a picture like this, like the 75 to 100 milliseconds, it could just as well be that this happens because prefrontal cortex sends information to the visual cortex. Because we know we have shown a stimulus to the participant, it's flowing from the eyes to the LGN to V1, but still it could be different. I think this is an important thing to say. So I think I've covered EEG quite in some detail here. I'm just going to take a look at the chat history again. Sorry, my kids are screaming. It's not ideal to be sitting here, but that's there's nothing to do about that. I'm going to stop the sharing again for 1 second. I'm just looking at this. Okay, I think I'm going to, pyramidal neurons are typically, Elisa asked it, typically projection neurons. They are able to project outside of the same brain region. And Noah asked, even though TESS is cheaper than TMS, why would you use this as it is imprecise? I think this really comes down to do you care about precision or not? So, for example, we know for example, in depression that we see a hypoactivation of the prefrontal cortex. If you don't know for sure which region you want to target, then you rather start with electrical stimulation because it's more imprecise and you don't know exactly which brain region to target than TMS. So there's pros and cons. If you don't know exactly what you want to target, then you don't want to use a precise technique. Is that clear?
Yes, very clear.
Okay. I think in the book, then AR, I don't know your full name, but they differentiate between a block design and an event-related design. I think this really comes down to what you want to measure. So if we're looking at the block design in the book, you can see, for example, you show a phase, then another phase, then another phase, then another phase. All these bold responses will add up, and at a certain point, they will reach a plateau. So you get a very strong bold response that you observe over, say, 20 or 30 minutes. And then afterwards, you show you have a rest block where the bold goes down to baseline. Then you show a house, a house, a house, a house, a house, again reaching this plateau in activity. So the bold response cannot go up further. And if you compare these two blocks, you have a very powerful design to say, okay, this region of the brain is more sensitive to faces than to houses, for example. But if I want to know where, like in a typical kind of working memory or decision paradigm, where you have all these different events happening quite soon after each other, and for example, if I would ask you to make a very easy math equation or a very difficult math equation, and that's the only difference that I'm asking you, then actually I have quite some events, perceptual, cognitive, motor steps that are in close proximity to each other. And then it more or less relates more to what we would normally do. And if I want to know something about mathematical aptitude and where this is, for example, linked to the parietal cortex, then I need to use this event-related design. I don't know, this is the conceptual difference between the two. So in the block design, we cannot isolate the single events anymore. And in the event-related design, we can.
Yes, in the book, they talk about how the PET can only do the block design.
Yeah.
Can't do the event-related design. And I wondered why instead the fMRI can do both.
Well, it's basically in fMRI, like, can you point out do you have the 4th or the 5th edition?
I have the 4th edition.
4th edition, okay, good. Our book.
And
can you point out the exact page number?
Yes, give me a second.
108, 109 or something.
Yeah, it's like the last, 108, I think. Yeah, block design experiment, yeah.
So if you look at figure 3.34,
yeah,
I'm just, that's the 4th edition. And let me just flip open the 5th edition here. Great. Okay, so what you can see in the block design, that you present the stimulus quite close to each other. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 stimuli. So say it's phases. In effect, all these, normally the bold response would add up, right? So if you space them in time, sometimes close to each other, sometimes further apart from each other, and you basically have different timings for when you present a stimulus, the first time, the second time, the third time, you could still mathematically get the bolt response back for each individual event. If you if you space them very close together and don't have any jitter there, so no variability in when you present them, then at a certain point the bolt reaches its plateau. So you cannot estimate for each single event anymore how big the bolt response for each individual event is. So if we want to link it to specific steps in cognitive processing or perceptual processing, if we don't have the jittering, then we cannot assess the strength of the bolt response for each individual event anymore. And you said something about animals or something like that, right?
No.
Okay.
Yeah, no, it was just this one, but I got it now. Thank you.
Okay. I think the block design is very suited for, say, perceptual paradigms where you want to see, does this region respond more to a face versus a house? So you overload it with lots of houses versus lots of faces, and then you get these big differences that you can also see in figure, I think 3.33 in the 4th edition is a very good one. And it's figure 3.31 in the 5th edition. So there you have moving stimuli and static stimuli. And there you can quite easily see that V1, for example, seems to care about both stimuli, whereas area MT cares about only the moving stimuli. So you see the clear difference there. If you would do that in an event-related design, not stacking up all the stimuli in close proximity, you probably need much more scanning time to get a big difference in the bolt signal. So here you actually want to maximize the bolt signal.
Okay, perfect. Thank you.
Good. Let's see, are there any measurement methods that do measure action potential, or is it always a measurement of combined input at the dendrites? From Iris, this is the last question I think that I'm going to answer today. Yes, sure, there are different methods to also measure the action potentials, but then you basically have to stick in an electrode into the brain directly. So if we're talking about healthy human subjects that we want to use for the experiments that we're currently doing, you always have to rely on EEG or fMRI, and then you're always measuring the combined input at the dendrites, either at a quite coarse level, if we're talking about space when using EEG, or on a finer grained level when using fMRI. So you can measure it with electrodes, but you have to lift the skull, and there's only exceptional cases where you can do that. So this is it for now. Thank you all for being here. Again, post your questions on campus. I hope this worked out. And I'm sorry that, well, we couldn't have any live lectures. It's much more fun to do it in that way, but there's nothing to do about that. So good luck in prepping everything. I hope to hear or see you again tomorrow.
Thank you. Bye-bye.
Bye.